of your species. Oh no.
We forgot to record from the beginning, but anyway.
Sorry, that was my bad. I was in charge of recording. Oh no.
You can't be in charge of too many things when you're, anyway.
Okay.
No problems.
Should I quickly re-explain this, since it's kind of, at least then if anybody else is gonna watch this afterwards, they.
Yeah, maybe for just this section.
Just this part is probably good.
Okay. Okay. Rewind like 3 minutes previous. Okay, so we're going to start with writing a simple model in the Antimony language, which is just a simple human readable language. Sorry for all those on the call, we're re-recording this. So we're going to go through this very simple model, which is a unimolecular reaction where we're expecting mass action kinetics. So the mass action rate law for this reaction is just the rate constant K1 times the concentration of the reactant species S1. So getting into the antimony syntax, we're going to define a variable ant string, which will be given the antimony string that defines the model. An antimony model is always going to be bracketed by 3 quotation marks on either end. And then we can get into actually writing the model. Start by naming your model. Basically here, we're just naming, we're declaring that we're going to name our model, and we're naming it simple model. The next step is to name to specify that to declare your species. So here we have species, which will indicate that we're about to declare the species, and then we're giving it 2 species that will appear in this model, S1 and S2. Okay. The next step then is to actually write the reaction. So here we have a reaction named J1. You can name your reactions anything here that is descriptive for the reaction that you're modeling, and then give it a colon, and so it's basically reaction name, colon, sorry, here this should be, okay, so reaction name, colon, write the reaction, semicolon, and then write the reaction rate law. So here we're giving it the reaction name J1. Our reaction then is S1 produces our S2 at a rate which is defined by the rate law, K1 times the concentration of species S1. Okay. So we've just defined our reaction where S1 is being consumed to produce S2 at some rate that's defined by unimolecular mass action kinetics. After you've given it your reaction in the model, the next step is to assign initial conditions to your chemical species. Otherwise, we won't be able to. Otherwise, the species will be automatically set to 0 when you try to simulate the model. So here we're giving some initial concentration to species S1, and to species S2, we're just setting it to 0. And you also want to assign constant values to the global parameters in your model. So here we're giving K1 some value of just 1. And then you can write N to end the antimony string, and you've basically just defined your simple model here. So we'll go ahead and run that code. Okay, so we've just saved our antimony string for this reaction as in the variable ant string. And we're going to go now and move ahead into simulating our simple model. And we'll get more experience going forward with writing the models. So hopefully it's clear the general format you're going to take, but we'll go through more examples as we go forward. Okay. So the next step is simulation. In order to do this, we're basically going to create a Roadrunner object instance, which we're going to assign to R here. So to do that, you're going to use Tellurium. I should have noted this, but when we import the package Tellurium, we're going to do essentially import Tellurium as T. So here, where you say CTE, we're just using some of the functions available in Tellurium, and that's going to be consistent throughout. So whenever you say TE. We're using Tellurium. All right, so the first function is Tellurium load A, which is loading to load an antimony string. And we're giving it the antimony string that we just wrote, ant string. So that will load your antimony string. It will create a Roadrunner object instance, which basically means we've created something that can be simulated, something that we can run these time course simulations on. The next command is really simple to simulate. So the command is r.simulate. So again, r here is your Roadrunner object, which contains your model that can be simulated. So r.simulate, and you're going to give it start time, end time, and the number of points that you want to collect over the course of your time course simulation. So it's going to simulate it, and then it's going to store your simulation result here as result. And then I'm just printing it here, which you already can see the output. So basically what this generates is it's sampled the system dynamics over time from time 0 to time 10, as I specified. It's taken 25 points of time, and it's reporting to you the concentrations of species S1 and species S2 over time. So again, you saw before that we defined the initial conditions for these species to be 10 and 0 respectively. So that tracks, that's what they are at time 0. Then once you have these results, you might want to store the simulation results in this format, but you also might want to visualize what this looks like so you can understand the dynamics of the system. So that gets into plotting, which you can do very easily in Tellurium as well. So we're going to go ahead and plot the results that we just generated. Basically, at this point, you've just run r.simulate. After you've run that, the results can be plotted using the command r.plot. So here we're going to plot the results of the simulation. So r.plot, we're going to give some title to our plot, which is uni mass action model. We're going to label the x-axis with time, the y-axis with concentration, because those are the variables for each of those axes, and just give it some fig size. And that generates this plot, which kind of shows you how these species are transforming over time. A little easier to look at than the tabular values. So again, species S1 started up at 10 and species S2 started at 0. And over time, species S1 is being consumed while species S2 is being produced. And eventually they reach some steady state value. Okay, so hopefully you have an idea. The main commands we've gone through now are te.load_a, which loads an antimony model, r.simulate, given that we've named our Roadrunner instance r.simulate allows us to simulate, and then r.plot allows us to plot the simulation results that we've just generated. Okay. If anybody has questions, maybe if somebody, you can feel free to interrupt me at any point if there's something I should add.
Yeah, Veronica, did you, I can't remember if you mentioned how to install Tellurium, because I didn't cover that.
I didn't mention, so I have installation instructions. I have a link to the installation instructions at the top of this notebook. But for this, there's a pip install. Did you do any, you did
No,
pip installs?
No.
Okay.
You might want to just type the command somewhere to show them.
Let me just, I'm sorry.
I put it on the Slack in the chat. But...
yeah, I'll just show you that block. Yeah. Oops. Sorry. No,
oh, I see you got it there. Yeah.
But it's really simple for this notebook. Really, the only package we have to install is Tellurium. So it's just pip install Tellurium. This just means don't generate all the output that you would otherwise be generated with the pip install. And then you're going to import the package, import Tellurium as TE. So
is there an exclamation mark at the beginning? You didn't mention that.
Yeah, there's an exclamation mark at the beginning.
Okay.
In Jupyter Notebooks. If you're doing this on your local machine, which we'll go over that on the last day of the course, just so if you're not used to working in notebooks or you would rather work in scripts, we'll show you how to various ways to install Tellurium on your machine, which you can do a pip install from the command line in most cases, but in that case, you wouldn't have this exclamation point, but you do need it for Jupyter Notebooks. Okay. Anything else that I should cover? Okay. All right. So we've gone through the 3 of the most important and also very simple commands. Now we're going to go kind of expand on that. So when you're working with a model in Tellurium, there are ways to basically set and report on values of either parameters or species within your model using Tellurium so that you don't have to continuously alter the Antimony string or go back to your Antimony string. So here I'm going to show you how to basically set a set one of the parameter values so that if you want to experiment with what happens if you change your parameter value in your simulation, you can do that without having to alter the Antimony string. So here I'm loading the Antimony string as before, te.loada and string. Then I'm going to do r.k1. So this is accessing the parameter value accessing the parameter k1. Gets 5, so I'm changing it. It was originally 1 in the Antimony string. I'm now changing it to 5, and then I'm going to run a simulation as before from time 0 to time 10, and I'm going to generate a new plot with these updated parameter values. So you can see that the dynamics in these 2 plots are different. What's basically happening is they're coming to a steady state faster, the 2 species, because we've just increased the rate of the reaction by 5 times. So these, the dynamics in the early part of the time course are much faster than in the previous simulation. Okay, so that's how you can set parameter values. One way you can set parameter values. You're also able to change the species concentrations in your model using a similar method. So here we're going to change the value of S2 to 10. So we're loading the model, we're changing species S2 value to 10, which in this case is going to change the initial concentration or the concentration before we actually simulate it to 10. And then we're going to run our simulation from 0 to 10 again. So I probably don't need to run these code blocks, but I'm just doing that so that if anything downstream has to be run, we don't run into any problems. Okay, so what just happened, we set the value of S2 to 10 instead of 0 as it was in the initial simulation. So now we see that the 2 species are basically, they have the same initial condition. And so the dynamics are a little bit different than before. Or at least that just the initial condition is different. The actual rate of the reaction is the same in this case. So you can access both the parameters and the species in the same way, and you can reset them without having to change the values in the Antimony string. Now, given that you've just changed some aspects of your model with Tellurium, you are going to want to pay attention to the resets in Tellurium. This is really important. And it's pretty easy to overlook sometimes. So make sure you reset when necessary. So here we're just gonna go back to our original simulation where we have the same load Antimony string command. We're simulating, we're plotting, and it looks just like it did before, exact same simulation. But let's say we want to change the value of K1 as we did before and rerun our simulation. Well, if we do that now and we don't reload the model, we're gonna basically, you're gonna start out at the beginning of the simulation here where you left off in the previous simulation. So you haven't reset the species concentrations to their initial conditions in this case, and you didn't reload the model. So you're starting wherever the last simulation you ran ended. So all the species, when you start this block of code, the species are at the final data point that was sampled in the previous simulation. So if we want to run multiple simulations in a row without having to reload our model every time, we have reset commands. So in this case, the simplest solution is just to use r.reset, which is going to reset the initial the concentrations of the species back to their initial condition. So here we're running the reset. which means species 1 should be at 10 when we simulate, and species 2 should be back at 0. And we get the same simulation as we did before with this updated parameter K1. However, that r.reset only resets the species condition, the species concentrations. So if you did change some of the parameter values, you're going to have to do a different reset, which is taken care of here with reset all. So in this case, we've just run this simulation above where we changed the value of K1 to be 5 instead of 1. If we want to get back to our original simulation, our base model that we created, we do r.reset all, and it will reset both the parameter value and the initial condition, our initial concentrations of the chemical species to their original state. We can run our simulation and we get back our original simulation results. So do keep the resets in mind when you're modeling. It'll definitely, it can cause, it will generate unexpected results if you don't reset your model. Okay, so the next important thing to mention would be that you can add events to your Antimony string, which are basically changes to the model that happen at discrete points in time. And maybe they only happen once, for example. So that's what we're gonna illustrate here. So we have this simple model with event. So we've just updated the model name here. It's basically the same model in terms of the reaction as the uni-uni mass action model that we were looking at before. Same species concentrations and parameter values, but here we have an event. So that's defined where we've named the event E1, and our event is going to happen when the time course simulation is greater than time 5. And at that point, the event that happens is that S1 is set to a concentration of 10. So basically you're adding a spike in S1 at time greater than 5. When time passes 5, sorry. So we're going to create the same executable model as before, loading this new Antimony string that we've just written, and we're going to go ahead and simulate that and plot it. And this is what the mass action model looks like with an event. So again, once the time passed time 5, you get a spike in S1, changes the dynamics. You can kind of think of this as like delivering a one-time drug application, which might have a discrete effect on the dynamics of the system. All right, so you can also set events. This is basically the same syntax for the model, except the event is different. In this case, we're setting an event to happen when the concentration of species S1 falls below a concentration of 0.005. And at that point, that's when we kind of add this spike in S1. So here you can see when we go ahead and run the simulation that S1 keeps falling, and when it finally reaches this very low concentration value below 0.005, it gets a spike and changes the simulation. If S1 didn't fall below that value during the sampling we have here, so between time 0 and 10, then you wouldn't see the event happen. Okay, so that covers events. Now we're gonna just kind of look at a slightly more complex model, which will incorporate interactions. So we'll look at the repressilator model, a very simplified form of it. There's a really nice form on the biomodels database. We'll talk about that a little later, which is really well documented. It has all the proper names for the chemical species. Here, we're going to basically just represent the species as proteins 1 through 3, which will be TET-R, CI, and LAC-I, and then also the mRNA for each of these to be M1 through 3. So those are the species that are defined in our model. They're going to have some more complex rate law, because obviously we're considering these repression interactions that are happening on the rate of production of all the mRNAs. So here you'll kind of see a pattern. Basically, these reactions are all split in either production of mRNA degradation of mRNA, production of protein, or degradation of protein. The activation and degradation of the protein is just going to be modeled using a mass action rate law. So just some basically production rate constant multiplied by the mRNA, the concentration of the mRNA will lead to the production of protein, and some degradation rate constant times the concentration of the protein will lead to the degradation. And also the mRNA will have a pretty simple mass action rate law as well. So their degradation will be modeled with a mass action reaction. But then you kind of see this more complicated term here, which is representing the effects of the repression that is happening from the other participating proteins in the network. And we're going to have some leak, which basically just means that even when there is full repression or very strong repression from the proteins, there's some basal amount of mRNA being produced all the time. Okay, after you've defined all of these reactions, we're going to go ahead and look at some species initializations. These values, they look crazy. The reason that they do, why they seem nonsensical, I guess, is because they've been optimized to generate oscillatory dynamics. They've been optimized to generate oscillatory dynamics, so their values are not necessarily biologically plausible. They're just mathematically able to generate the dynamics that we wanted. So each of the species is initialized to some value, and then all of the, sorry, this should not be variables. This should be like global constants or parameter, I should say parameter. These are the parameter values for the model, and all of them have been assigned by the end of this model string. So we've written some model that should be able to generate the oscillatory dynamics of the repressilator. We're going to go ahead and load that same command. Repressilator gets te.load and repressilator string, which is what we named our antimony string. Then we're going to go ahead and we're going to save this as an SBML file. We'll talk more about SBML a little bit later, but it's basically a standardized format, and it's really useful for exchanging across platforms. It's one of the, it's a different type of model. It's a different type of model description format. So if you think of Antimony as one way to write a model, SBML is essentially a standardized way of encoding a model. And so we're going to save our model as an SBML file. And this is just telling you to get the current state of the SBML model. Again, we'll go into this a lot more later, the standards specifically. All right, then we're going to run the simulation, simulate from time 0 to 100, 500 time points, and go ahead and plot it. And so we were able to generate these oscillatory dynamics using the three species repression network that we saw in the paper in the beginning of the lecture. So that's nice, but how can we like look at this model in more depth using Tellurium specifically? So some more functionality that you want to consider in Tellurium are some of these functions that allow you to actually look at the species, at the parameters, and their respective values. So here I'm just going to go ahead and print the floating species IDs, which are just the variable species in the model. So the mRNA and the protein concentrations and the names of those species. I'm also going to print the global parameter IDs, which are all of the parameters that we defined in the model. So all the kinetic rate constants, the leak constant, and we'll also print the values for those. So once we've printed them all, of course, we're getting all the species names. So this is a good way if you're looking at somebody else's model and you've loaded it, you can print the antimony, or if you're just interested in knowing the species that are in the model, you can use this command instead. Okay, so yeah, it's just going to print there's obviously a lot of parameters in this model, so it's printing the IDs of these, and then this long list here is going to be all of the values for those parameters. You can do the same. You can also print the species values as well. Okay. So these are some pretty important functions to consider. One final comment I want to make, and then we'll go into exercises. I think we're doing hopefully okay on time. So like I said, we'll go into the standardized formats a little bit later, but I did want to just comment on the utility of using Antimony. Hopefully you understand the syntax. Happy to answer any questions after about the syntax, but it's pretty easy to read and write Antimony, whereas SBML, which is the standardized format, is not really, it's not, especially for large models, it's not really easy to be digested just by looking at the string. This is just showing you, using that get current SBML command that I used earlier, what this repressolator model looks like in the standardized format. All the same information is there. It's in a format that can be exchanged across platforms, which means that you can use other simulators, for example, to run some of the same simulation experiments, but it's not really easy to understand what's going on from this model just by looking at the SBML. Whereas if you do the same with the Antimony string that we wrote, you can, it's organized in a way that's very easy to interpret as a human. So just a reminder of the utility of using the Antimony language. Yep, so this is the Antimony version versus the very, very long SBML format. And the great news is that Tellurium allows you to export these standardized formats after you've written it in the simple format. So very helpful. Okay, that's pretty much the end of all the simple tasks that you can do in Tellurium, unless Herbert or Lucian, you have any some more of the simple commands that you would want to include here rather than later that I can demo. But there are exercises, so hopefully you can start kind of working on some of these tasks and just get used to actually writing the code to do it, which is probably the most helpful way to learn it. There's answers to all the exercises too, so.
Yeah, the only thing someone was wondering about in the chat was what turned out to be assignment rules. Like, can you set something to be the cosine of time? And you can with a colon equals, that which is called an assignment rule. Is that?
Yeah. I might not be able to write a fast assignment rule, I think, but maybe I could.
Yeah, just like if you can make up a new thing, just S20 colon equals cosine of time or something is easy enough.
Let's go to some previous model.
Yeah.
Yeah.
Oh yeah, I see what you're saying. Oops. Is it just cosine or is it cos?
COS, yeah, parentheses time.
Now, CS0, you won't be able to control a variable. It has to be a boundary species.
It should it'll automatically do it. But
Should I just set S2 to that instead?
I don't know what'll happen there.
Then
oh, never mind.
Yeah, make it a new one. S2, then S1 isn't actually creating S2. It's just always fixed at the cosine of time.
I mean, that would do anything because
say
that's
species
S20
S20 at the
not
top.
connected to anything.
Oh, okay. Well, I didn't define the.
You don't have, like, so if things appear in reactions, they're automatically flagged as species, but S20 doesn't appear in the species anywhere, so you have to say species S20. There you go. And then it should be in the.
Right.
There you go.
Okay,
Yep.
so here's S20 down here. Yeah, there's a limited number, or like, I mean, Lucian, you obviously is the expert on this, but you can implement pretty simple functions, piecewise functions, using this syntax, and that should be in the Antimony documentation. All the different assignment rules that are available in Antimony.
So Lucian, if you do this, then does S20 automatically become a boundary species? Does it?
Yeah.
Oh, okay.
if you were to like round trip that back into Antimony again, it would come through with a $sign by it, which we have also talked about. But
yeah, that's one thing I'll cover tomorrow.
Yeah.
Boundary species.
But yeah, anything you give, and I guess the other thing would be a rate rule. Like basically, if like reactions are sort of the bread and butter of SBML and Antimony, but if you need to say like this thing changes in time at this particular rate, you can just say S21 prime with a little accent with a apostrophe after it equals whatever your rate rule is. Here, so like that or something. And that would just create a species that, and then you need to say what it started off as.
Yeah.
And then it would just go up with time. S21 would be exactly the same as time because it changes at the same rate time does.
Yeah. Okay.
We probably have like 15 to 20 minutes to work on exercises and for people to ask questions if that works, and then we can take a 10-minute break or so.
Sure. Are we doing breakout rooms and stuff?
Okay, there's the question. You can unmute and go ahead and ask.
Sure. Oh yeah.
Oh, sorry. Yeah.
Do you want to ask?
So I had a confusion while resetting the parameters. What if 10 parameters are declared and we want to set 9 parameters out of those 10? So it's quite cumbersome if we list all those 9 parameters. Is there a way to reset on, like we list only the exception one? Like we list that we reset all the parameters except for the 10th one, because in that case, if that 10th parameter is supposed to be updated in the future steps, so we need to keep that updated version instead of resetting it.
Well, you could either reset all of them and then change the one that you wanted to have updated, like re-execute the one command to change the parameter that you want to stay fixed at that value. Or you can loop through, you can create like a list of parameters that you want to change. Like you could loop through all of those parameters and basically reset them to their initial concentration using like looping through 2 lists where you're saying, okay, reset this parameter to its original value. Then you don't have to write all those commands out necessarily. I think those are probably the 2 easiest way. I don't think there's a, but I think the first, if you're trying to just keep one parameter updated, then probably the easiest way is to store that update in another variable, reset your model, and then change that parameter back to whatever the update was.
Oh, okay, thanks.
So do you want to do exercise 2? That seems the most basic one to try out. I think people should try to implement this.
Yeah. I mean, most of them are pretty easy. Yeah, definitely start at exercise 2. The first one, you don't have to do this. There's just another, I think Herbert mentioned JWS Online as well. It just might be something useful to consider if you want to look through some models and simulations that already exist, just as like an exploratory exercise. But to actually get familiar with using Tellurium, start with exercise 2. And most of them are the pretty simple commands that we just went through.
So if anybody hasn't got it installed, let us know. Need some assistance.
So we had a question about the S21 prime syntax. If you say S21 apostrophe prime equals something, equals 5, then S21 will change in time, like it'll set D S21 DT to whatever you say on to 5. Or you could have a complicated mathematical equation there, whatever you like. If you have S21 in a reaction, then every reaction that appears in the ds dt that it will calculate will be the sum of all every time it was created in any reaction, every time it was destroyed in every reaction. So let's see. So okay. Anyway, so yeah, you can set it directly to say like, this is how it changes in time. Don't reuse any of the reactions, use this thing instead. Yeah, so yeah, probably if you could copy your thing in there or share your screen or something, I don't know, either one's fine. Or should we go ahead and do breakout rooms now? That makes sense?
Yeah, that might be better.
Okay.
Oh yeah, okay. Veronica, do you want to send, what are you going to do?
I don't know if I guess I can.
I can do it.
Do I have to, how many breakout rooms do we want?
There's 53, so maybe 6 or 7.
How many people, how many?
Six will assign about 8 to 9 people per room. So if people either want to work together, yeah, I guess that would probably 8 to 9 seems reasonable.
Yeah.
I think. Okay.
And then should we have TAs visit the rooms periodically, or?
Yeah, we'll visit them then.
Okay, I'm gonna go ahead and assign.
Should we pause the recording now?
You can basically, you can place in brackets basically the species you want to collect data from. So here you want to specify time because this will allow you to still do the r.plot command and plot the species concentrations against time. And then I'm just going to collect data from species S2 so that those are the only selections that you have in the model. Yep, and then in that case, this r.simulate command would return a result that has time points sampled and also concentration values for S2. And then when you plot the simulation results, you just get the S2 concentration. Yep, so you could do the same. You could add in species S3 here and just leave out S1 using the same syntax.
You can also put in parameters or the fixed species or something.
Mm-hmm. Or the reaction.
Or the reaction rates themselves, yeah.
Yeah. Okay. If there are no more questions, we'll take a 10-minute break and then we can get into the second half of the session.
I will say the one thing that came up in my breakout was the Antimony syntax for when you name a model. You have to say model, the model name, and then your content, and then you have to end it with the word end. That is unlike Python, where the indentation is all you need. In Antimony, you need the word end to sort of wrap it from the beginning.
Okay, 10-minute break. We'll meet back here soon. Thank you. Everybody can see my screen fine. Should be good.
Yeah.
Okay, sweet. All right, so this lecture should be a little bit shorter than the other ones, I think. We're going to look at kind of data collection, so which databases might be relevant to the types of problems you're investigating, and how do we access that data, and then also model annotation, which has to do with taking metadata, or basically information about the measurements we're collecting, and incorporating that data in our model to make them more descriptive and allow others to better understand what it is that we're modeling. Okay, so, and then the exercises we'll go through, it'll kind of be more about, it's probably most useful to you if you try to get an idea of these databases and what kind of data they have available to you for your specific project, but also there are some exercises that you can do that are like predefined and a little bit guided. Okay, so what is data collection? This is just a process that's necessary for any modeling project where you're taking data from multiple experiments, potentially if you're able to collect it from experiments, but also you're going to scientific papers and you're taking data that has been previously published, and also you're using online data sources that in order to ultimately fully inform your model to the best of your ability. It's essentially trying to get as much information you can about the system before, in order to guide your model to be more biologically relevant. So typically you would need to curate your collected data to kind of ensure that the quality of the data you're collecting is acceptable for your model, which might involve eliminating maybe data that's recorded for a particular molecule you're interested in, but is taken from an animal or something that isn't really relevant to what you're studying, or maybe it just doesn't quite meet your quality specifications. So what databases are useful then? There are a lot of databases that you might want to use in your modeling workflow. Some of the main ones are going to be SBRK, which basically stores biochemical reaction kinetics information. So it'll incorporate information about what species are involved in the reaction and the rate constants that have been measured for those reactions. And it's heavily focused on metabolic and signaling network reactions. So we'll go through some examples of using SBRK. Actually, I can give you a quick, so I just have pulled up here the SBRK database. It has a pretty simple search infrastructure. You can give the molecules you're looking for information on. You can give the tissue you're looking to collect from. So here I'm looking for reactions that involve ATP and are the tissue they're recorded from is the brain. So I'm going to get this list of a very long list of reactions that have those search terms. You can really make this much more targeted using the advanced search option. But then you can kind of try to curate this. Which reactions are useful to your model? Are these reactions you're planning to model, or are maybe they related, but you're simplifying some aspect of your model so you don't need to incorporate that information? But this will basically give you information about, it'll give you identifiers for proteins that are involved in the reaction. It gives you conditions it was recorded under. So here are these wild type variants. Potentially there would be, I don't know, if you're looking at, maybe if you're doing a study on metabolism in the brain in the presence of some brain cancer, maybe there's a cancerous variant that you want specific kinetic data for. And then this has the organism it's recorded from, the parameters that they have information about, and even temperature conditions that that the data was recorded under. So all of this could potentially be useful for your model and might be relevant to include. So yeah, but SBRK is good for actually giving you information about the kinetics. Brenda is good for information about enzyme catalyzed reactions. So it'll basically classify the information in the database based off of whatever reaction that enzyme catalyzes, and it gives you kinetic information about the substrates and the products. ChEBI is a dictionary of small chemical compounds, so small molecules basically, and is probably relevant for a lot of the systems that you may be looking at. KEG is gives you genome information and also pathway information, so it'll have like full pathway maps. I have one pulled up here for mitochondrial respiration. So any pathway that involves modeling mitochondrial respiration is going to come up with some pathway information here. You can go ahead and like look at these pathways. I think I have one pulled up for the longevity regulating pathway in a worm, and you can see the you can see where the mitochondrial respiration processes are happening, but this is basically a much larger map in which it's incorporated some information about our search term that we're interested in. But maybe we're interested in modeling this full map, and this can kind of inform the interactions that we include in our model, or maybe we just want this subset of the map that's focused on the mitochondrial respiration aspect. But it gives us kind of a starting point to understand what different players are involved in our networks. Biopsych is a collection of pathway and genome databases as well. So there you can search for genes, proteins, metabolites, and pathways, and basically the occurrence of your term will be looked for across multiple databases. So it basically is good for querying many databases for sort of generic terms that could be incorporated in a lot of different databases. And then the biomodels database is the only model database we're going to talk about in this course, which we'll go into much more detail about later. But this actually is a repository of biological models, the types of models we're talking about in this course that have been published in literature, and many of which have been curated so that so the results have basically been validated by a team of curators. And it can be a really useful starting point if you're modeling some system that's already been attempted before, like maybe you can build off of existing models. And then I just have an appendix here from a paper which provides a very comprehensive list of data sources for intracellular biochemical data that might be relevant. So this is by no means an exhaustive list. There are many, many databases that might be useful to you. And of course, they're going to be, depending on the domain you're working in, very specified databases that could be useful. Okay. Yeah, so just in addition to that, we're going to be obviously focused on the computational aspects of basically what can we do with data that's already available. Actually measuring data for experiments is a highly collaborative process for many modelers, so it's not necessarily possible to always gather all the data we want with our own experiments. So sometimes we have to make simplifications based off of the data that's available. And of course, we'll talk a little bit about parameter fitting tomorrow, so you'll kind of get an idea of how to work around maybe not having all the correct data that you would ideally have. All right, so this gives us an idea about data that we're going to want to look at, but what is metadata and how much of that should we collect? So metadata is data that describes biochemical data. That's how it's always described, which maybe isn't the most useful definition, but basically it's giving us information about the identities of chemical species we're taking measurements from. It gives us unit information for parameters that we might be measuring. It could give us estimates of measurement accuracy. So if we have if we're given a mean value, does it give us an information about the standard deviation around that mean, so we can incorporate accuracy, we can incorporate uncertainty into our model. This can help us, it can help us write annotations for our model. So basically incorporate this information, incorporate this metadata, so we have a more descriptive model, sorry. And ontology terms defining the annotations. Basically, ontologies are just a standardized form of communicating about a given topic. So we'll go into some of that later in the course as well. And then, so you'll collect information about all of these things. You also want to collect provenance data. Provenance is kind of, it's a specific kind of metadata which describes basically the history of that data. So where it was collected and when, and it's the conditions it was collected under, and then also any manipulations that happen to that data, like post-processing on the data set, would be incorporated in provenance data. So here, yeah, I also have, you might want to have the protocol that was used to generate the data. And you could also incorporate identifiers for the paper that actually was published with that data set incorporated in the findings, just to give more information about where all of our data sources are actually coming from, so that can be checked by other researchers. Okay, so that's just kind of a short overview. We're going to get into kind of how you can actually access some of the data programmatically. Some of these examples are going to be simpler than others, so don't get lost too much in the in some of the more complicated Python aspects. When you're importing data programmatically, there's often a template that's going to be available. Depending on the database you're collecting it from, they have their own web services sometimes that'll help to make that an easier process or not. Bioservices is a Python package that allows you to programmatically access many of these databases, including KEGG, the PDB, which is the protein database, and KEBI, so the small molecule database. So we'll go through some examples of that. It's a lot easier than some of the other web services Python programming. So if you are able to use bioservices, it's a pretty good option. And then we'll talk a little bit about annotation at the end. Okay, so bioservices is going to allow us to basically query or search for some quantity or identifier that we can use in a model. You could potentially import a kinetic rate constant, which we'll do later, and use that to inform our model. So you could directly set the value of that kinetic parameter in your model after you query the database, basically. And ideally, by querying programmatically, you're creating something that's reproducible. So somebody else can just run your code and they can return the same values that you were able to get before. And it eliminates a bit of human error that would come with basically typing in, going to a database, searching, and then just returning, basically copying and pasting some value into your model. Definitely that's something that is done commonly, but if it's possible to make it reproducible, that's a good choice. And doing this programmatically at least helps that a little bit. Okay, so here we're going to have an example of using bioservices, and we're going to programmatically search the KEGG database. So here I guess maybe let's go back up this. So this is hidden because there's going to be a lot of output that's pretty messy, but just to go through some of the packages. Okay, so we went through all these pip installs. I told you in the last session, we went through what a pip install does to get Tellurium. And then we have imports for the packages that are going to be relevant to this notebook. Bioservices specifically, we're basically importing all the functions with this import star. That's just taking all the possible functionality, the classes available in bioservices, and it's importing them with whatever their name will be. So some of these are going to be like KEGG down here is actually getting imported with that command. So that's where KEGG came from. It's basically it's available through the bioservices Python package. All right, so KEGG is good for looking up genes and genomes. So we're going to go ahead and look up the TETR gene, which is one of the genes that was involved in the repressilator circuit that we went through before. So basically, I'm creating basically a database in Python. So I'm assigning the KEGG database here to the term database, to the variable database, and then I want to basically search that for some value. I'm using a basically KEGG ID number. So this is something that I would have to know, or I would have to see it in somebody else's model, or I would have to have searched KEGG for this term. But this identifier is unique to TETR, to the gene TETR in the KEGG database. And I'm now going to basically search the database using this database get function. And I'm returning the information about this search to TETR query. So again, query here is just going to be whenever I do some search, it'll be called query. Using that query, I'm going to build a dictionary. We won't go into a lot of detail about the dictionaries right now, but this term database.parse. The query is basically returning a dictionary that can be then searched for various different keys. So dictionaries, you can inside of this dictionary, this TETR dict that I have here, you're going to be able to search it for name, which is going to return basically the name of the gene that I've just created the query for, and definition, which basically just provides a definition for what TEDR is. So that I'm searching the dictionary that I've generated using the terms name and definition here, which both come with some associated value in the dictionary. Okay, so that's printed out here. I should probably just run this through here. Yep, I'm not sure why this is coming up right now, but this is the first print statement. It's giving me the name of the gene that I've just returned from the database, and also it's giving me a definition, which says it's a TEDR ACR family transcriptional regulator for the tetracycline repressor protein. So yeah, it's given me some information about this identified gene. Pretty much all of the bioservices queries are going to look somewhat similar. We'll do another one a little bit later, but first we're going to go through an example of importing a kinetic constant from SABio-RK and why this might be useful to do. This is where we get into a little bit of, I guess I should ask first if anybody had any questions with the syntax for querying the KEGG database. Or if you do, you can post in the chat and I'll try to answer that. Okay, but for the SABio-RK search, there's not a bioservices. Bioservices doesn't provide access to the SABio-RK database, but SABio-RK basically has its own guide for accessing data programmatically. Oops, I shouldn't have done that. There's a spelling error. For accessing data programmatically, which you can find on their website, but basically what I'm going to do is first give some URL. This is all defined in their documentation, so I can point you in the direction of that after the course. But basically, you're providing some URL to a query that you want to go to that website. So it's as if you were going to launch SABio-RK in your browser, except we're going to do this programmatically. So it's using the Sabio REST web services. So yeah, it's still going to launch. It's going to try to access information that you would otherwise have to go to the website to look for. You then basically, it has a special format for creating a query dictionary here, where I'm going to be searching for basically information related to the spike protein in SARS coronavirus binding to the receptor. So I'm going to specify an organism, which is SARS coronavirus, and then I'm going to specify a product of the reaction, which will be this spike glycoprotein complex, which, let's say I know that this complex is necessary for SARS coronavirus to infiltrate a cell. Therefore, I'm interested in modeling that process of the SARS-CoV-2 virus being able to infiltrate cells. So I want information related to this particular reaction. Again, this is going to be some specific syntax for Sabio's RESTful web services, but what it's doing is it's essentially just joining these with an AND statement. So it's going to be searching for the organism SARS coronavirus and the product of this spike glycoprotein. Yeah, this is just unpacking that. Then you're going to specify what fields you want to include in your include when you access the database. So I want to return information about the ID. So if there's some standardized identifier in SABio-RK, can return that. Tell me which organism the data that's returned was from. Hopefully it's all from SARS coronavirus, because I've included that as part of my search query above. Then incorporate, add the UniProt ID, which is going to be for the complex that I'm interested in. Standard identifier, and information about any parameters related to that product that I'm interested in is going to get returned as well. This gets a little bit clunky, and it looks like it's not showing up. If you're able to run the notebook in a Jupyter notebook later, it should show the full table here. But hopefully for now, this should be sufficient to illustrate the point. But the next, after you've basically finished your query, so now we know that we're looking for a specific organism, a specific product in a biochemical reaction, and we're going to go ahead and return information related to these different fields that would otherwise show up in SABIORK. So you kind of have an idea about some of the fields that can be returned here, like organism here, parameter here is what we're going to be returning primarily. Okay. These are standard. These are where you actually are making the request. Basically, the RESTful web services are going to look at the website and return information about your search terms. Then we're going to go ahead and just print some information from the results that we get. So this response should basically incorporate all the information we're getting back after we've gone and we've accessed the information on the website. So I'm just going to print the results of this search. There's some processing done to convert it to a string here. Then you want to convert it to a CSV file that can be read into a data frame. Data frame is basically a really efficient way of storing and storing data and accessing data. It's very clean way of organizing your data set, and it's pretty standard in Python. I think we'll get into more details of using the data frames later on, but for the case of this, for the sake of this, I just want to be able to basically print and visualize the results of the search query that I'm doing, which I'm going to do here, and that's what we're getting down here. So once I get that query back and I get information from SABIORK, a lot of these in these columns that are left out, these are basically incorporating the other terms I've asked for. So the Uniprot ID is hidden right now, and some other parameter terms are also hidden right now. But it is returning a few of these entries that are in the query. It's giving me the SABIORK entry ID, the organism, and then I'm getting the units here. Presumably there's also another parameter here. For example, parameter type would tell me well, what type of parameter is. For example, is there a rate constant in somewhere in this query? Parameter start value, which we're going to search for here, is going to tell me what the actual value of that parameter is. And then parameter unit is actually displayed here, but it's just telling you what unit of measurement the experimental data was recorded in. So I'm going to go ahead and search these essentially to try to store information about this reaction that I'm interested in. Okay, so we have our data frame here, and if we want to just know information about the parameter type, I'm just selecting the parameter type column here using the name of the column, which would show up here like this parameter unit as well in the data frame. And then basically this is going to have a list of parameter types. So presumably one of the types in this list is rate constant, which is what I might be interested in if I'm trying to define how fast a reaction happens in a model. So I'm going to search for rate constant in parameter types, and if that rate constant shows up, I'm interested in knowing information about the start value and information about the units of that value, of that recorded value. So I'm going to store those as complex formation rate constant and complex formation rate constant units variables, which I can potentially use later on. And so then I just have a print statement here where I'm going to print out the rate constant for this reaction and their units for this reaction, which is getting printed here. So after querying the database, I can find that the formation of the spike glycoprotein complex occurs at a rate of 0.112 seconds per, inverse seconds. And this is potentially information I can then use in a kinetic rate law for a model, which we'll go ahead and do in the next step. This is going to be basically not really based on necessarily the the model we're going to look at is just something I wrote to sort of represent a system, but it's not really going to be biologically relevant because I don't have a picture of the figure of what we're looking at in the notebook. Let's just look at this quick example here. So what we're trying to understand is basically how the reaction where the spike glycoprotein on the virus binds to the receptor on the cell membrane, and that's it, just that reaction. So essentially just this S1 RBD subunits in green binding to this ACE2 receptor is what we're looking at. And the query that we just did found information about how fast that happens. So the rate constant, I returned a rate constant. Now the modeling component that we're going to do then, obviously we don't have, there are probably models that exist currently that have a better representation of that reaction, but here, just for the sake of building on what we learned in the previous lecture, we're going to just make a reaction where we assume that there is mass action kinetics. So here we have the two species. We have the receptor ACE2, we have the spike glycoprotein RBD and the subunit 1. And then we also have the complex that forms as a result of the reaction. So then I've named this reaction complex formation because that's the process that's happening in this reaction, which has two reactants, ACE2 and the spike glycoprotein, and those react to form the complex ACE2 spike complex. And we're saying that it happens with mass action kinetics. It likely doesn't, but just as a example, we'll have some rate constant K multiplied by the concentration of the first reactant and the concentration of the second reactant. And that will be our full definition for the reaction. And then we can have some not biologically plausible initial conditions for the receptor and for the spike glycoprotein. And we'll also have some parameter value that we assign to the rate at which this reaction happens. Now, the utility of the query that we just did in SABRK was to basically try to find a rate constant that is more biologically plausible for the system. So we're gonna load our antimony string, which is named spike protein binding string above. And then we're gonna set the value of the parameter K, which is the rate constant for this reaction, to the value that we actually curated from the SABRK database. So that was saved as complex formation rate constant here. And then presumably you can go ahead and you can simulate your model with that rate constant. Obviously, the results of this model aren't going to be very biologically meaningful because these values are just picked to be able to show as an example and aren't based on the true system. But this is the utility of trying to aggregate information about a given rate parameter and use it for your model. And programmatically, once you've aggregated that data, it's really easy to update your model parameter values and try out simulations that incorporate that. Okay, so I know that this kind of gets into, sorry, I've missed a lot of the chat. If anybody has questions, I can take a quick break now, but this antimony model, I just wrote it by hand. It's pretty short. Yeah.
Is it possible to get the antimony model from the database that you just downloaded?
So there's no antimony. The database that I was looking in has information about the biochemical reactions, but it doesn't actually have a model to represent those reactions. So some of them might have information about the rate, like for example, I imported a rate constant. So I tried to find out the value of this K here, hypothetically, but I don't necessarily know that this is the rate law that the reaction follows. And that information might not be in the reaction database. It might just have information about the rate constant.
But how about the list of reactions? Like, even though if you don't have the rate law, but knowing what is the list of reactions might be helpful, right?
Yeah, there should be ways to import the list of reactions. It might not be in the most intelligible form necessarily, but you could import it in a way that like you won't be able to probably automatically generate the antimony string from that, but you can at least use it to guide your building of it. But as far as I know, you'd probably have to do most of the antimony building by hand. Maybe Lucian can answer that. If that's
I can say, like, so these websites aren't going to know about antimony ever, but they might know about SBML. You might be able to find something that will export SBML for you. And then you can load SBML into Tellurium just like you load antimony into Tellurium and with a load S instead of load A. And you can get, once you have the SBML, you can convert that using Tellurium or Antimony's converter from SBML to Antimony, so you can look at it and stuff. I'll have an example of doing that tomorrow if you want.
Yeah, and for the question about how to add values that are more relevant, really that's just doing this process many more times, I guess. So you might be able to find, maybe if you're studying, if your model system here is like the human lung and you're looking at the virus infiltration in some cell type in humans in your lung, then you would want to find information about how concentrated are the spike glycoproteins on the coronavirus, and how concentrated are the ACE2 receptors on the cell type that the virus is binding to. But this all takes a lot of expert biological knowledge and a lot of searching of databases. So data curation can be a pretty time-intensive process for sure. Okay, I'm going to go through one more example, I guess. This is just using the bioservices API again, which is much easier to use than SebRKs, I would say. But we're going to do the same. We're going to try to get some metadata, basically. So not necessarily a actual experimental value, but information that would describe maybe a biological term. So here we're going to try to retrieve an entry for D-fructose 1,6-biphosphate, and we're going to search the small molecule database KEBI. So again, we're going to create this database object that is searchable, queryable, using the KEBI database. Then we're going to create a new query for this database. So we're going to get the complete entity for the value of this KEBI. This is basically a KEBI ID. So whatever molecule corresponds to this KEBI ID. These IDs are unique to a very specific biochemical species. So it's only going to give us information about one distinct biochemical species. And then I'm just printing out some of the information that I can get from my query that I just retrieved. For example, I'm printing the definition of this small molecule, and also I'm printing out the KEBI ID, of course. We know what the KEBI ID is because we just put it in, but presumably you can also look up there may be other IDs associated with the molecule that you could also look into. So this is just giving me a definition of the small molecule I've looked up, which could be useful to include in your model, or you can actually incorporate the KEBI ID itself. And we're going to go into that a little bit in the annotation section, although I don't think we have a KEBI annotation. But so if you haven't heard about model annotation that much, it's basically a process of trying to incorporate metadata in your model description so that the components of your model can be identified by somebody, by some independent researcher with extreme specificity. So they shouldn't have to really guess at what the biochemical species are or at the experimental conditions in your model. The models we've used so far, specifically the simple antimony model, have been very simplistic representations of like common mechanisms. So mass action kinetics, but if you see something called species S1, you don't really know anything about that in terms of a biological system. So it's not a descriptive name. It doesn't give us that much information. And it would be really hard to understand if we were actually modeling a true biological system. So we want to be transparent about where the data that informed our model originated, and we want to be as clear as we can about what the components of our model actually are. So we're going to go through a quick example of actually adding annotations to the repressolator model that we wrote with Antimony. So there is a way to add some simple annotations directly to our Antimony string that we didn't talk about in the last section. So we'll do that here. This is all the same repressolator model that we showed in the previous session. Now, here we're using really non-specific names, right? Like M1, P1, and then all of these kinetic constants we don't really know. Did that again. The kinetic constants, we don't really know what they actually are. We don't actually know which, like, of the 3 repressor proteins they are related to. So you could add information through annotations about these. We're not going to add specific annotations about the identities. Well, here we'll add information about the protein identities just as an example, but you can add a lot of different, there are a pretty large number of possible annotations you can add here. Here we're just going to add some SBO terms, which is the systems biology ontology. It's basically the place to go if you're trying to find, some terms to describe what processes are happening in your model. So we'll go and look at one of the examples from the systems biology ontology that I'm incorporating in the annotations. So I've incorporated this term 356, which corresponds to some decay constant. So we can gather from that just by understanding what the exact identifier is, that the reaction, this D_M1 is what this rate constant is doing, is that it has something to do with the decay of a biochemical species. So this is true for all of these same parameters. So I just incorporated that. And here, this SBO term is going to correspond to a Hill coefficient. So if I look that up, it's going to be the SBO term for Hill coefficient, which means that we can assume that our parameters that have n in them in the model are all Hill. All 3 of these parameters are Hill coefficients. And then these are a little bit more descriptive of the exact repressolator system. But if we look up what the identity is of P1, we get that it is TEDR. So we know that was one of the 3 proteins in the repressolator circuit. Okay, so what we've basically done in Antimony, there are a couple of different annotations you're able to add. One is the SBO term or other similar, SBO term is one of the main annotations you can add. So you just give the parameter name dot SBO term gets SBO : and the exact systems biology ontology ID that corresponds to that. So that's one option, which is what I did for the decay constants and the Hill coefficients. And then here, the protein identities, you can use, basically the syntax is parameter or species name, its identity is, and then you give a URL to the exact identifier for that term. Here, since they're proteins, I'm giving you identifiers in the UniProt database. So that's this P04483 is the exact identifier for TEDR protein. And this is a pretty simple way to do it in Antimony. Now, these don't really cover actually that much that is happening in the model. There are a lot of other parameters like these leak constants, or, we have degradation, decay constants, but what about these constants that correspond to the production of a biochemical species? You can add annotations for all of those. This is also a pretty time-intensive process generally, but taking the steps to add these annotations is going to make this model much easier to understand. If all I have is what I originally showed you in the first session, you can have an idea about what the system is capable, the dynamics the system is capable of. You can have an idea, you can understand information about how the species are connected based on the reactions, but you don't get information, but you don't know anything about the biology of the system really. You just have all these, ambiguous names. So adding the annotations can really help clarify what you're modeling. In addition to doing them in Antimony directly, there are some other options. One, which you'll hear about in the Symposium session, is this package SBML utils, which can make it which has various functionalities, but one of them is to add annotations to SBML models. So here I'm gonna load this repressolator string from before. I'm gonna save it as an SBML model so that I can annotate it. Basically, SBML utils is only compatible with SBML, with annotating SBML. So the simulation, not necessarily necessary, just to remind you that this is the repressolator model. Oh no. Okay, this might not work. I probably just hopefully missed this, I guess. Hopefully that's the case. No, that's not the case. Okay, well, I'm not sure why this is not running now. I'll have to try to fix that and figure out what's going on.
And just do a run all real quick.
Let's try that. Hopefully. This might take a little bit, but I can basically go through really quickly what happens, how you do the annotations using SBML utils. Basically, I'm going to be providing it here. Here, I'm just taking a data file that I have uploaded to GitHub. So this is going to be a CSV file that contains all the annotations I want to add. If you're doing this on your local machine, you can just write an Excel file, basically some tabular format, tracking all the, okay, this is not working. I'm pretty sure there's some package issue, so I'll have to fix this, and I'll upload a working version and let you know on the Slack what the issue was with this. Let me see if I can try to bring it up in a different format, maybe. Just for the sake of demonstration. Okay, hopefully this works properly. There are a lot of warnings here going on. I think some of the packages weren't very compatible with the Google Colab environment. Let's see if these run. Sorry, it might take a little while. I can walk you through the code just in case it doesn't run properly. So here I'm just giving a file. This is, like I said, if you have it on your local machine, basically if you have the table of annotations that you want to add, here it's stored in a CSV file, but you could do it in an Excel file too. SBML utils is compatible with a couple of different tabular formats. Yeah, so here I'm basically using this class model annotator to read into a data frame, to a pandas data frame, the annotation file that I'm specifying. So right now it's going to pull it from this GitHub URL. It's going to pull in the CSV file, and it's going to create a data frame called repressor annotations that contains all the annotations that I have curated on my local machine, created a file for. And then after that happens, here's just like a print statement to show. Basically you would create this CSV file while you're modeling. It would be kind of similar to programmatically trying to access all these databases. It's just a different way of storing the data. I don't know if this is gonna run properly, but I'll just open. It's taking a while, so. Okay, this is basically what the file that I'm importing looks like. And it's what SBML utils takes as input. So you have a column that specifies the pattern that you're looking for in your SBML model. So these are going to be the rate constants and parameter and species names that I've specified in my repressolator model. So SBML utils will search for that pattern in a string, and then it will add some of these annotations. So I'm specifying what SBML type it is. All of these are considered parameters in SBML, in the SBML format. Then the annotation type, all of these are going to be RDF, which is just a specific format for adding annotations to SBML files. Actually, I'm not this is a standard qualifier. I'd have to check what it means exactly. I think it is just saying that this parameter D_M1 is, and then whatever term is specified in resource. So it's saying D1 is SPO term 356, which, as we showed before, is just a decay constant. So this is just some additional information. I don't think this gets directly, it's going to be directly output. But this is the data file that's getting read in, or is supposed to be getting read into the notebook, which doesn't appear to be working right now. It seems something is, oh, sorry. Yeah, there's a, okay. I see what's happening. This is a older version of the notebook that's uploaded, so it might not work, but it would be great if it did. Okay, great. Okay, so this is just printing out the data frame that I just showed you. So yeah, this is basically what it looks like if I want to print out a data frame instead. I was showing you an Excel-based file, but it basically is exported as CSV and is included in the repository. Okay, so then in SBML utils, you're going to go ahead and actually start the annotation. So that is done with the command annotate. You're giving some source, which is the original SBML file, which is your model without the annotations, or with some annotations that are incomplete. Annotations path is just providing it the file, the annotation file that is printed here. And then file path, which is going to be whatever you want to output. So it's going to be your new SBML model with all the new annotations included. So I'm taking a source, I'm adding annotations, and then I'm generating a new SBML model that has all the updated annotation information. Okay, yep. And then after that's done, this gets saved as repressolator doc, which I can convert into an SBML document. These are commands that are just part of the SBML utils commands. It's going to create this repressolator annotated SBML, basically string. Yeah, it's going to be an SBML string here, and then I can use Tellurium save to file. So that's another function that's available in Tellurium where you can specify the file name that you want to call it, and then you can save the model as that file name. So I'm saving this annotated SBML model to repressolator annotated XML. And then if I want, I can go ahead and load that model that I just generated. And I'm printing the antimony for it so that it'll be human readable. So yeah, load SBML model, repressolator annotated that I just created. And then I'm going to go ahead and print it as an antimony file. And now you see I have these SBO terms and these identifier, species identifier terms that just got imported. All of these were just imported from this file that I talked about. Yeah, I think that's all that I have for this notebook. I'll have to figure out why this is not running properly in Google Colab, so I'm really sorry about that. Most of the exercises then are going to be, sorry, I have all the solutions open, so feel free to take a look at the databases and get an idea for how they relate to your specific project. I think the last couple of exercises, the last exercise will at least allow you to try to query a database programmatically using the bioservices API, which was the easier one, kind of has a pretty standardized format of how to query them. And then also to try to add annotations to an antimony model, which you can just do by editing the antimony string. I know I missed a lot of questions, so that's pretty much all I have for the time being. So if there are questions that are pressing, if somebody wants to ask, otherwise I can go back.
There was one question earlier on that was about like specific Python syntax for you had like a query, like and in quotes dot join %s, and it was like, what the heck is going on here? So
yep. So basically what this line would do is if there were, right now there's only these two like items, this query underscore dict items. The items are organism and are the values associated with organism and the values associated with product. You could have a much longer query dictionary here if you want to incorporate more information. For example, if you have a specific Uniprot ID, maybe you could add Uniprot ID and some value. But what this line is going to do is it's going to go and it's going to take all the items in your dictionary and it's basically going to join them with this and. So it's not really that common. I guess most databases can be queried with pretty with like simpler language now, but often the queries will add like in like a easy to understand by humans way will get translated into having this like and, which means we want all of these values that are in the query dict to be incorporated in our end query. So yeah, this is just taking.
Can you print out what the query string is just so we can see what happens? Sure.
Yeah, it should be the first print.
There it is.
Yep. So pretty much all it did was it added this and between the two.
And then the %s colon %s is where you get the organism colon SARS coronavirus and stuff.
Mm-hmm.
It's a compact bit of Python code there, but yeah. And this is also, I think, people's first introduction to a dictionary, which maybe so a dictionary is a way to map an index to a value. So as long as all the indexes are unique, then each index will be associated with a different value. In this case, our indexes are strings. So our two indexes in the query dict are organism and product. And then the value for the organism is SARS coronavirus, and the value for the product is the string spike glycoprotein, blah, 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 blah. You can make, so if you had query dict bracket quote organism, the result would be SARS coronavirus. And dictionaries are pretty common in Python to store information by index of some form or other. Like the simplest form of index is 0, 1, 2, 3, but you can have strings or you can have anything else that is unique and can't be changed. That's a really quick introduction to dictionaries, but yeah, there we go.
Yeah, this is what's getting returned from the print statement. So I'm giving it the key organism and it returns a value, which is SARS coronavirus.
Yeah, dictionaries are also referred to as having key/value pairs. So you have a pair of things. That's the other bit of syntax that is being used here that I mentioned in the chat is the parentheses k comma v for k comma v in query dict items. Loops through query dict dot items, which gives you pairs of things, and then assigns them to the first element of the pair to k and the second element of the pair to v. And then the join syntax. Oh no, I guess it's the syntax with the %s colon %s quote percent parentheses k comma v puts k as the first %s and v as the second %s, and then what's in k of v is defined in the next bit. So yeah, there's a lot of things going on.
Yeah, there's a lot going on. Definitely don't get afraid of that. Even like, I guess, so like this is definitely a template that SEDRK provides on their website, and I don't think, I mean, yeah, this is pretty complex. I guess most people probably wouldn't certainly
don't expect to write
don't
that
be afraid. Yeah,
freehand.
don't expect to
when
write
you're a
it.
beginning
No.
Python user.
No, yeah. This is just modified from a tutorial that SavvyLRK has. Probably this would not be the first way I would think to write that line of code. It's pretty complicated. So yeah.
All right, we have a question on chat. Do you want people to solve the exercise 1 through 3 by Google search, or do you want to write code for it? Or
you're welcome to do either. If you're excited to write code for it, go ahead, for sure. You can also just, if you want to explore the databases to some extent, most of these are just to try to get you like familiar with the databases themselves. If you have specific, if you have specific entities that you're interested in for your problem, definitely substitute that out and you can do those. I think we have about 20 minutes to work on some of these. And then, yeah, I don't know, Herbert, if you want to rejoin 10 minutes before the hour or 5 minutes before the hour to go over an overview of tomorrow or something?
Yeah, sure, yeah.
Okay.
Yeah. I just have to remind myself what we're doing tomorrow.
Do we want to break into breakout rooms again? It's kind of more, it's less focused than the other exercises, I would say.
Yeah, we can stay here and then people can just send things on chat and so on if they have any problems. Okay. And if you have any other questions in general about what we've done today, now's your chance to ask as well.
I would say people do feel like in the breakout rooms, people are more likely to feel free to chat about, like actually like share their screen or chat. Feel like do be open for that. Or if you want to have a breakout room with a TA, that we can do that also. If you don't want to share your questions with the entire class, which can be a bit intimidating.
Yeah.
I can just generate breakouts. That's okay.
Okay then. Is it 10 minutes, is it?
Yeah, 10, 15 minutes.
Okay.